So, pleasure to be here and talk to such an interesting crowd of people with varying backgrounds. I'm a professor at the Department of Infectious Diseases, but I have a background in environmental science. So I've sort of tried to merge environment issues with research on antibiotic resistance. So I'll have the opportunity today to talk about the environmental dimensions of antibiotic resistance, but also the interactions with the animal compartment. And we call that the One Health perspective, which I'll talk a little bit more about that later. And you're, of course, invited to ask questions at any time. I'll do my best to reply or answer. So, I hope you can see and hear everything, and we'll go to the next slide. So what is a One Health perspective? Well, it's usually referred to as the interconnection between human health, animal health, and environmental health, actually. But when you talk about antibiotic resistance, it's rather about how human health and environment and sort of animal health is interconnected with the environment. And this interconnection is clear if you think about how bacteria can move. Bacteria can move between, not only between people, but also between people and animals, and between people and the environment, and back again. And it's not only the bacteria themselves, but also the genes that can make bacteria resistant to antibiotics, that can flow between these different compartments. And for that reason, it's recognized that you need to sort of address all these three different areas in order to manage this global challenge more efficiently. So, I will not really talk about the human disease compartment today, but a little bit about the animal part and the environment and how these two contribute to the overall resistance problem. So these so-called One Health approaches, they are stressed in a number of important documents that lay out strategies for how we should act to combat antibiotic resistance. You see here the World Health Organization's Global Action Plan that delineates much of what they think is needed to be done. Here you see the Swedish strategy to combat antibiotic resistance. And actually every country is supposed to develop such a strategy soon. Some countries, like Sweden, have a pretty strong environmental compartment. Others barely mention it. So it's a lot of about the degree of awareness, I think, among those involved to whether there is a true involvement of the environmental compartments. So I'll start talking about the animals. Oops, this one. So animals, why? Well, we use antibiotics in animals, you know that. And we do that of several different reasons. The obvious reason, of course, is that we need antibiotics to treat infections in domestic animals. We can't really stop that. It does not make sense from an animal welfare point of view to not treat sick animals. So this is something that is largely considered necessary, but of course it differs how you do it. It's also necessary to use antibiotics sometimes to prevent infections during outbreaks. If you have a farm and a disease starts spreading, antibiotics is an important tool for you to prevent even more animals to get sick, and which will not be good for your business and will not be good for the well-being of the animals, of course. What's more controversial is the use of antibiotics for routine prevention of infectious diseases. Because this is quite commonly applied actually in many parts of the world, that you're giving, on a regular basis, you give animals antibiotics so that they won't become sick, thereby increasing your economic benefits, of course, of the business, but with a drawback that this may drive antibiotic resistance. Another fourth use of antibiotics in animals is to promote growth. Because low doses of antibiotics, certain antibiotics, will actually increase growth of several different types of animals. And of that reason, antibiotics are used as growth promoters in many parts of the world. Not so in Sweden anymore, but we used to do it as well. But in the majority of the world, it's actually used. And this is also a very questioned use of antibiotics. And I'll come back to that a bit later. So these are the four uses: treat infections, prevent infections when you have an outbreak, or routine prevention, or growth promotion. And when you farm animals, crowding, you put many animals together, that will increase risks of transmission of diseases between the animals, of course, because they get in very close contact with each other. And by using antibiotics in such settings, one could actually squeeze animals a bit closer together than what you could do otherwise. And this is the reason why it has been or is used so much. So alternative to this is to work on hygiene conditions in the farms, whether you farm broiler chickens or pigs or whatever. If you create systems that prevents transmission of disease from outside and into the barns, or between the animals, or between subgroups of animals, you can do a lot to reduce the need for antibiotics in the farming situation. Now the next thing I mentioned also growth promotion for animals, and just mentioning that sometimes it doesn't need to be the critical antibiotic that you use to treat humans that can drive resistance, but it could be other similar antibiotics that could drive the resistance. So in this picture, you see a very complex molecule called vancomycin. It's a critically important compound to treat certain types of infections. For example, during neonatal care. So this is an antibiotic that we should be really, really caring about so that we don't drive resistance. But for many, many years, this very structurally similar compound called avoparcin was used to routinely feed chicken and pigs, also here in Europe, to increase their growth. And what was discovered is that the bacteria carried by these animals developed resistance to avoparcin, but resistant to avoparcin also means resistance to vancomycin because these molecules are so similar. So it took quite a few years, but then this was recognized, and now we are not using avoparcin anymore, as I understood it, in Europe as a growth or for growth promotion. So this is good news, of course. But if you move out further, you'll find that other critically important antibiotics are still used for growth promotion. There is one antibiotic called colistin. It's a very old antibiotic, and we actually stopped using it many years ago because it had such nasty side effects. But with increasing problems with multi-resistant bacteria, we had to go back and see, is there anything that we used to use that could be useful? So colistin has become an important drug again for treating the worst kinds of infections. However, colistin, this is regularly used as a feed additive for chicken in India. They get it with the water. And just a couple of years ago, the first mobile gene that can move between bacteria that provides resistance against colistin was found in, I think it was China in this case, it wasn't India, and it was found both in farmed animals and in people. And now we know that it's spread all over the world, basically. So this is a kind of use that I think the world needs to think more about, of restricting. So how does it look like with animals and antibiotic use in Sweden? Well, we use more antibiotics to humans than we use to animals. But in a global perspective, it's probably the opposite. But we are pretty good in not using so much antibiotics for the animals. And if you look at how this has developed over the years, you can see, I think, quite an interesting pattern. These are sales of antibiotics to animals in Sweden, and it starts here in 1980 up till now. And you see different colors there, where the purple one is antibiotics used for growth promotion. The green one is group treatment, where you treat the whole herd of animals. And the blue one is when you treat an individual sick animal. And you can see here something important happened here between 1984 and 1986. That was when Sweden banned the use of antibiotics for growth promotion in animals. And of course, we obey laws here, usually. So they stopped this kind of use. But of course, there was still quite large proportion. I should say that this stop of using antibiotics as growth promotion in Sweden in 1986 was also proposed on the sort of the EU level, but it took until 2006 until it was banned in the EU. So 20 years to convince the other countries here that this was not a good idea. Now they are discussing it in, for example, the US. But it's still used, not to talk about how widely it's used in countries like China, for example. You can also see here that the green bar, the group treatment, goes down. This means that we have basically increased the transmission control in farming, so that we don't need so much preventive treatment of the entire groups, but we can actually focus the treatment on individual animals. And that has brought down the use quite a lot. If you, on the other hand, look now how it looks like in different other countries in Europe, I mean, we have maybe 10% or less here group treatment. In the rest of Europe, it's almost the opposite. So they have primarily group treatment still, and much less individual treatment. And overall, they also use more antibiotics. And this you can see on this graph here. Here you have different countries in Europe on the x-axis. And on the y-axis, the height of the bars, you see how much antibiotics they use for the animals expressed as milligram per kilogram live weight of the animals. Approximately, this is what it is a measure of. So if you treat big animals, you of course need much more antibiotics than if you treat small animals. And what can we see from this one? Well, we can try to find Sweden. Here is Sweden. We have a very small bar. So we have been pretty good from an international, or very good from an international perspective, in keeping down the use. There are some other countries that are doing really, really well. You have Iceland over there and Norway. One reason why Norway is so good, you know what animals they're producing in Norway? Salmon is what they produce a lot. And they used to use quite a lot of antibiotics in the salmon farming. But they did something really radical some decades ago. Do you know what? They started to vaccinate each individual little fish when they were small, basically eliminating the need for antibiotics. So if you can vaccinate against those common diseases, it's a super solution. It doesn't drive resistance, it prevents disease. Problem is that it isn't that easy to develop vaccines against many bacterial diseases. But it has worked very well for the Norwegian salmon farming, which is a great success story, of course, from many points of view. You can also see from these slides that it's different what kinds of antibiotics we are using, but some classes of antibiotics that I think you have maybe touched a little bit upon are dominating. You see the dark blue bars are really important. That's the tetracyclines. And also the light blue bars are quite important, which is the penicillins. In Sweden, we primarily use penicillins. And the primary use is actually to treat mastitis, juverinflammation, in cows. Because cows are big and they require a lot of antibiotic if you treat them. So, for example, for chicken, we hardly use any antibiotic at all in Sweden. Is Greece not there? Why is that? Cyprus is there, and that is a bad guy, I think, from this perspective. I don't know why Greece is there. From the top of my head, I think that Greece is a rather large user of antibiotics. Overall, you can see a quite typical pattern. If you move from north to south, you see that the antibiotics use is increasing. And this is a pattern that you also see if you look at how common antibiotic resistance is among people. You see a clear pattern. The northern countries have less problems, and the southern countries in Europe have considerably bigger problems. And it's very much related to the antibiotic use. If I talk about antibiotic use in humans and plot that against the resistance level, there's a reasonably good correlation actually between the two. It shows that, yeah, antibiotic use in humans is a major driver for resistance. So tetracyclines, whether it's used for certain animals or certain types of species or infections, the answer is yes and yes. So it depends, of course, on what animals you have. For example, in fish farming, tetracyclines are really, really important if you don't vaccinate the fish and you don't need antibiotics at all, for example. So it matters depending on which kind of infection and which kind of animal you do. And this is probably the explanation why, partly an explanation why you have a difference. But it's also sometimes a matter of regulation and general tradition and practices. So if we continue that question and look at how this use is divided by different types of animals, you see here, what animals do we use the antibiotics for? And it's quite striking that half of the antibiotics we use to pigs. I guess that's primarily as growth promoters in China. It's probably the biggest part of this pie chart. Then it's chicken, and here we have shown that it's possible to eliminate basically the use of antibiotics in chicken farming. Our chickens are a bit more expensive than those countries that might be a bit more expensive, but not so expensive. I still think personally that chicken meat is very cheap. Our pig meat is a bit more expensive than some other countries, clearly. And then it's cattle, sheep. And of course there's also use for domestic animals, but that's smaller, like dogs and cats, etc. So we're using more and more for our domestic animals. Domestic animals are more and more treated as family members, and if they get sick, I mean, in the old days you shot them, but that might not be the thing today. You bring them to the vet and you pay a fortune to treat it, quite commonly. So this is driving a use for more and more broad-spectrum antibiotics also for domestic animals, actually. Again, when we look at the different classes, you see that it's quite different types, but tetracyclines and penicillins and quinolones are big. Quinolones is a bit scary because they are broad-spectrum antibiotics that are known for driving resistance on a broad basis also to other types of antibiotics. So now I come back to your question about South America. So this is how it looks like if we look at the antibiotic consumption for animals, for animal production, I should say, in 2013. And then it's a little circle there how it's projected to increase in the years to come. And it's quite striking where we have the highest use of antibiotics. Can you see the dot in Sweden? No, you can't. It's really small. But China is dominating, clearly, followed by South American and North American countries, right? And then, of course, there's quite a lot of use in some of the European countries as well. So this, it's not very minor, but really it's the growth-promoting use here that you see here that is the biggest part. Yep.
If you correct for the number of animals produced.
Mm-hmm. That is what was done in the previous slide here for the European countries. So this is estimated per kilogram weight, but I don't have that. I don't have that data. Maybe it's there, of course, but I don't have that data for China and for the US, etc. But as you can see here, here is adjusted for animal weight, and it's huge differences between countries. You see Cyprus, for example, here. That's, by the way, if you buy these rubber cheese things, what do you call them? The halloumi. So good, right? Almost all of it comes from Cyprus, where they use this much. Do you know why they use so much antibiotics there? Because they have a system where the veterinarian earns money if he sells antibiotics. It's a really, stupid system, because it's a really good incentive for prescribing lots of antibiotics. So you really need to get rid of that kind of systems in order to reduce the use. This is not, of course, done here, and this may be one reason why we have a low usage also. There's still incentives, actually, for a veterinarian to prescribe, because if the veterinarian, or if we say a doctor that meets a patient, meets a patient, animal or a human, they want antibiotics, right? The owner of the animal wants the animal to get antibiotics, and it's easier for the doctor or the vet to prescribe antibiotics than not doing it. But adding that the vet then actually will earn money on that of course makes the situation worse. So I go back, and let's go from the antibiotic use to the transmission process of how bacteria are moving from animals to humans, because they are. We get in close contact with animals, not always as close as on this slide, but they can spread. This is not, don't practice this at home. Isn't that what they're saying? Folks. So, if we look at a bit more boring slide, I illustrated it like this. So this would illustrate the possible pathways, main possible pathways, how resistant bacteria could transfer from farmed animals to the human population. So we could have sort of via the environmental route, so the farmed animals, you have particularly fecal residues from their poo that contains lots of bacteria that goes into waste, maybe it's spread on farmland as a way to reuse nutrients. It could come and contaminate our fruits and vegetables. We know that we sometimes get sick from eating strawberries, for example, that has been sprayed with sewage to just fertilize the ground. and you can get different kind, often viral diseases, but still you can get also E. coli bacteria, etc. from that way, and they could be resistant as well. That's one possible way. We could have contamination of basically food products that are made by animals, such as meat, etc., or eggs. Particularly if not care has been taken how the slaughter process and the food process line is handled. Oh, that's this one here. We could, how did I mean with the animal feed and byproducts? This was very clear to me when I wrote it, but at the moment I don't remember. So I'll see if I can come back to that when I think about it. I was thinking of that one. Probably meant something with that one, I'm sure. So at least that one and this one here. This is direct transmission from farm workers and food processors, those that come in contact with the animals. And then they transmit the disease to the population. So it's not you as a, in this case, it's not you as a consumer of the food that gets transmitted, but by buying the food that is maybe produced with a lot of antibiotics, you will promote this route of spreading, so somebody else will get it, and then it could spread in society. So now, different bacteria spread in different ways. And some species spread very efficiently from animals to humans, and some not. Salmonella and Campylobacter are two bacteria that we quite easily get from contact with animal or animal food, basically. Like eggs or chicken is one particular case with Campylobacter. And you know that you need to be very careful when you handle chicken meat in your kitchen, for example. MRSA, you can get that from, for example, pigs. You can get it with contact with dogs also. There's a related species there also that the dogs tend to carry sometimes that is also resistant to methicillin. And then ESBLs. Did you talk about ESBLs as well? Okay. So intestinal bacteria that have become resistant not only to penicillin but also to the next generation, basically the cephalosporin class of antibiotics, which usually make them very difficult to treat. And there's really good evidence for the top one, a little bit less for the latter one, for the ESBS, how the interconnection is between spreading from animals to humans. So what can everyone do about things like this? Well, you can eat more vegetables, of course. For most people, it's probably good to eat more vegetables, I guess. But it's, in my taste, it's a bit boring. So I'd like to eat meat myself. So if you want to do that, you can, of course, think about what products you're buying. You just think about buying animal products that are produced under good hygiene conditions and with the minimum use of antibiotics. Thereby, you, as a consumer, will not contribute more than necessary to the spread of antibiotic resistance. And as I mentioned, at home you can always practice good hygiene also. These are some simple things that anyone can think of to make a change. Those were the parts I was going to talk about with regards to animals. So if you don't have any specific questions about the animals, I'll move on to the external environment. Or we can save it for later also, of course. Okay? Then let's talk about the external environment and the environmental dimensions of antibiotic resistance. And I'll talk about largely three different roles of the environments. I will talk about the environment as an arena for the evolution and emergence of new forms of resistance in pathogens. I'll talk about the environment as a transmission route where bacteria can spread, but also the environment as a sentinel or as a reflection of how the resistance situation is in the local human or animal population. Because relating to the first point here, if you look at the environment, it very often tells you something on what's happening in the sort of the human compartment. This environment here is very dirty. It's not dirty by itself. It's because humans has been there and done something to it. And you can do this in a quite elaborate way. When it comes to measuring chemicals, you can figure out a lot of our practices by studying chemicals in water. For example, some colleagues of mine in Italy, they studied residues of cocaine in the sewage treatment plant of Milano over the different days of the week. And the pattern they find, they look like this. So much more party in the weekends. No big surprise. It was a similar study in Sweden where they followed actually cocaine residues also over the time course of a raggy festival in Uppsala. You can see that there were clear patterns there and basically a footprint from the raggy festival. And this same team, they actually also did a similar study where they followed cocaine residues along the River Thames that flows through London. And they found the highest levels of cocaine just downstream from the Houses of Parliament. I think that reveals a lot, right? Or maybe it was a sewage pipe that came out there, who knows? But I think it was quite funny that they found it. So basically, looking at the environment can tell us stories about the human situation, and that is possible, perhaps, also if we look at the bacteria there. So we have a research project here in Gothenburg where we're looking at sewage bacteria to try to say something about what the resistance situation is in the human population. This is an idea we have. We don't know whether it will actually work, but it could be quite interesting because if you can do that, it can be useful, maybe not so useful here, because here you have pretty good surveillance data on how often, when a doctor should treat a patient, they know that, well, the chances that this antibiotic works is this and this much, and that this antibiotic will work is some other percentage. Because we collect data all the time from patients, so we know that. If you have an infection with an E. coli, you know the likelihood of treating it. And you need to know that as a doctor, right? You need to know that when a patient comes in because you may need to treat immediately, long before you know what the resistant pattern is of that bacterium. But how is it in Africa? There's no surveillance in most countries there. The doctor has no clue what will work more than his own experience. And that is our idea here, that if we could, in a cost-efficient measure, go there and look at sewage and provide information on what is actually the likelihood that certain antibiotic would work, that could be an idea. So this is just a research project ongoing, but I think it's quite exciting. And we'll see in the future if this might work. But I think there is a role of the environment here as information channel for how it looks like in the local human and animal population for certain bacteria. Cocaine was just an example. It did not have anything to do with microbes. It was just to illustrate that what we do will be reflected in the environment. So the bacteria that we poo out, that will be reflected in the environment. So if we go to India and look in the rivers there, we find a lot of bacteria resistant to last resort antibiotics. But if we do it in a river here, we don't find that. And that is, of course, because the resistant problems is so much bigger in India overall, this is fecal bacteria that we find there, than it is here. So we sure there is some signal there. The question is how accurate is the signal. Okay? So that was the sort of how the environment can reflect the resistance situation. So now I'll start talking a little bit, oops, wrong computer, about the role of environment in transmitting bacteria. Contaminated water is recognized as a route for spreading bacterial pathogens. Here, we are pretty well off with clean water. We rarely get sick, although it happens, from drinking water or from swimming in water or coming in contact with surface water. But it happens, of course, here also. But in some parts of the world, this is standard, because in many parts of the world there is a complete lack of sewage infrastructure. In India, there are hundreds of millions of people that don't have access to a toilet. They poo on the ground. And maybe in some places there are toilets and collection systems, but there's no treatment. It's just a collection system of sewage, and it's released to waterways. And in these regions, people are often poor, and many of them live like this. So no surprise that this is a way of how we spread pathogens, including resistant pathogens. It basically makes them circulate like this. But one thing I'd like to stress here is that it's not only the spread of resistant bacteria through contaminated waters that drives resistance, but actually the spread of any kind of infectious agent. Let me explain that. If you are, particularly if you come from a low-income setting and you get sick, you buy antibiotics, regardless of what you get sick from. Even here we want antibiotics regardless what we are sick from, right? We go to the doctor and ask for antibiotics for many things where it's not needed. So infections caused by non-resistant bacteria or viruses or protozoa or something else all contributes to the overuse of antibiotics. So if we can invest in improved hygiene in these areas, that would do a lot for spreading disease in general, transmittable disease in general, but also lower the use of antibiotics and thereby also lowering the accelerating development of resistance. And we had stressed that in a paper of ours some time ago also. Now, waters and contaminated waters are critically important, but we also contaminate our soils with waste. So we know that spreading manure from animals or sludge from sewage treatment plants is a good way of bringing back nutrients. So we have a sort of a closed cycle, and this is very attractive. We use it. I mean, bringing manure on grounds is practiced all over the world, has been as long as we have been practicing agriculture, I guess. And we're also bringing back from our sewage sludge, primarily to circulate phosphor as a resource. But of course, this means that we also spread antibiotic residues and, if we're not careful, also bacteria. Here in Sweden, we have a system that eliminates most of these bacteria that could cause disease by a special heat treatment, etc. that does this, so that makes the sludge less risky of spreading. But if you go out and look at soils that have been sampled and stored since the 1940s until now, this is in the Netherlands, you can see that while the older soil samples contained basically no resistance genes to these classes of antibiotics, you see that the newer samples, they contain a lot. I took away the slide that was about how much antibiotic is left, but it depends on the different kinds of antibiotics. Some antibiotics are really, really easy to degrade, like penicillins, for example. They are fragile molecules that basically are, even when we pee them out, they're almost then they're gone to the largest extent, whereas others come out in basically unchanged form. Think about those that you use for urinary tract infections. They need to come through the body in rather unchanged form because you want them concentrated in your bladder, right? So sulfonamide antibiotics, fluoroquinolone antibiotics, they're pretty stable, and a large portion of those come out with the urine or the manure. So you have a portion that comes up through the poo and some portion that comes out through the pee. Basically, and this differs. Since it's easy to measure the pee, it's difficult to measure the poo chemically. Anyway, so but these authors, I think they primarily interpreted this that, oh, we have an increased load of antibiotics here in these, they are selecting for it. But it could also be, I argue, that the animals from which they collect the manure, their intestinal flora looked very different in the 1940s compared to now. So it could be basically that the manure contained little antibiotic resistance then and much antibiotic resistance now. So it might not be a result, or I don't think it is a result of the antibiotics in the soil. I think it is a result of the use of antibiotics in the animals over this period. But it results, what you can see is that these genes are there now in these environments. So this is a change. So soil is a potential source for this. Now, we've gone on for about 45 minutes, and time flies. So I'll take a break here.